Thank you. So I'm Jeannie Martin, I'm a clinical pharmacologist, and I've come to this field quite late. And really I've come to it because of a clinical need. So working as a general physician in Queensland, I had a large number of cancer doctors who came to see me to say that they prescribed the patients that they had the right gene test and they had been prescribed these drugs and their patients either weren't responding or they had significant toxicity. And they wanted to ask a pharmacologist what they should do with their patients. Should they reduce the dose, should they double it, should they move to another drug? And actually we had no answers to any of the above, despite doing large amounts of research. And hence started a long journey which is involved with many of the speakers here today. I think we're just embarking on really, which is to try and really essentially replicate into the cancer genetics area or cancer therapeutics area what the immunologists have done 10 or 15 years ago. And it's probably actually even more difficult because you have to go back to the original hypotheses and the original aims and try and sort of take away not just the hype of the genetics revolution and what this can do for cancer, but also the strong input we've had from the pharmaceutical industry. And there may be pharma people here and don't have anything particularly against the industry. In fact, I work quite closely as a pharmacologist. But I think we have really had a lot of money and pressure from us to adopt new drugs without really understanding the biology of the cancer, the different genetics of the tumour as opposed to the human host, and what's actually a new mutation and what's actually a germline mutation. And even more importantly, I think, as a pharmacologist, which is how do these drugs actually even get to the tumour in particular patients? Is the pharmacology the same if a patient's old or if they're young, or if they're obese, or if they've got comorbidity, or if they're on drugs that, for example, inhibit 2D6 or inhibit BRAF or KRAS from some other mechanism? So really we're kind of starting off a low base, and we've got a lot of, we're sort of really against the tide, so that's why we have to be very careful about picking off the bits that we think we can make a scientific contribution in. But I do think this area is quite timely because we are having difficulty trying to get enough patients to do clinical trials in all these patients that have specific gene mutations. We see that when we evaluate our work for the Therapeutic Goods Administration, that these trials for new drugs have got smaller and smaller populations with smaller and smaller subsets of genetic mutations. So I think that is becoming a problem from the regulation perspective. It is also becoming a problem from the funding perspective because we know the PBS, the Pharmaceutical Benefit Schedule, is really under stress here. In fact, it's actually not sustainable when you look at the trajectory of what's coming and what we have currently committed to fund. And I know that there are very high-level government discussions at the moment of the new chair of PBAC about to be announced with huge money about to be committed to cancer drugs. That we're really starting to think about this whole paradigm of gene mutation here equals X drug here at this one dose. So I think it is quite timely, but more importantly, I guess from a clinical perspective, that we've got these rafts of patients that have got severe toxicity from some of these newer agents. So they might be targeted in that they are known, well at least we know they've got one mutation, but they're so specific that they actually block that enzyme in every single bodily system. So now we're actually dealing with patients who don't just have neutropenia and hair loss, but they have major life-threatening and mortality-causing side effects from some of these new genetic therapies. And in addition, because we're going down that track, we're actually not really thinking about how we can use some of the older drugs more effectively. So I think for all of those three reasons, of regulation, funding, and safety and efficacy, it's probably timely just to sort of put a hand up and say, hang on, we know the genetics is really interesting. I know it has the promise of really being able to help us get the right drug to the patient. But there's a whole lot of pharmacology we need to understand about whether, for this particular patient, this drug at this dose is actually the right thing to do. So I've kind of been a bit cheeky today. I must dissociate myself from my poor university, who I think just put up with me. And this is really my own views and from the people I work with, some on the front panel, but not the views of the government I work for, the university, or the pharmaceutical industry I consult for. And really, the idea is around how we can individualise dose in the real world. That is, patients that are not in the clinical trials, real patients that are 90 years old and are having treatment for their hematological malignancy, along with their renal failure, their diabetes and their hypertension. And whose response is more to just a gene mutation that's in their tumour. Their response, as we've already heard, is not just a gene. It might be the concentration of the drug that's actually there at the cell, which may be related to their metabolic genetic pathway, or it may be related to how much fat they have, and therefore what the volume distribution is and what the concentration of that drug actually is, and also how long it takes to actually clear the drug. And that's actually really important for drugs that have to have a certain amount of drug at a target for a period of time. And we've seen that with some of these cancer drugs. So I think I've sort of got my future cut out for me now. I don't think I've got time to do anything I was doing. I think I'm now caught up in this pharmacology mission, really, I guess, which is to say that there is a lot of work for pharmacologists to do, but we can't do it on our own. We need to be working with scientists. We need to be on the ear with policymakers, with other clinicians, with our colleagues, and saying, hang on, have we actually thought about the pharmacology principles here? So I hope I'm not going to bore you today, because as you can see I'm quite passionate about pharmacology, but I do think that the principles of pharmacology are really important for everyone to know, everyone who's playing with therapeutics. And I'll try and make it a bit interesting by putting a pharmacology example for each of the drugs or situations that we bring up today. And then of course if you really want to hear pure pharmacology, you can tune in now to our new Newcastle pharmacology curriculum, which we're actually about to put online. And that's got all the nuts and bolts of pharmacology together with all the mathematical and academic esoteric things. Okay, so this is like a big amount to get through, but a lot of it we can get through pretty quickly. I want to just start off just by raising the question of how we got here. How did we get into this, what I think is a bit of a mess, really? We've got this huge patient demand, this huge society demand for new drugs to keep coming in, to be funded and registered, and all the safety data, and these competing demands of making sure that we're not going to make the patient sicker, we can actually afford it, we're not taking drugs or funding away for other areas of the health budget which might actually deliver much better patient outcome. We don't want to make our elderly patients so sick with these cancer therapies that they end up dying in hospital, as we've seen, and should be perhaps able to die at home if they knew about the option of having these adverse events. Maybe talk about a few examples. The very few examples I said where we know using genetic knowledge to guide dosing improves outcomes, they've actually probably pretty much been covered already. Abacavir, Carbamazepine, there's 3 or 4 where we know that information can really improve health outcomes. That's not to say the others don't, but we just don't have the same level of post-hoc validation in a randomised controlled study of patients with the gene or not getting a specific therapy. And these factors, which are really pharmacology issues, we know there's all these phenotypic factors that affect the relationship of a drug or a genetic test to the outcome, and we've already seen that there are ethnic reasons why people with the same gene might actually express it differently or actually have a different response to a drug. And then I thought, rather than just sort of, try and be smart and say we've got all these problems, which I think a lot of people are probably already aware of, we actually have to do something about it. And it's really trying to think of both a clinical and academic methodology of saying, well, these are where we think the biggest problems are, we need to address those, or these are where we think the biggest gains are to be made, and how can we set up a framework that can do this. And I don't think that one person sitting in Newcastle University can do anything about this. I think that this is really a collaboration that has to occur between people that are representing the speaker group and in the audience today. And I don't think that we can be precious about taking bits of it necessarily and trying to, well, we can take bits of it, but I don't think we can take ownership of the whole area. It really is a collaboration and big minds on the job. And then I've just got some mathematical ideas if we have time to talk about how we're trying to deal with these little snippets of genetic information to try and improve dosing. So we do have some information on some drugs that might help particular population groups, and we're using some mathematical modelling to try and predict how that might be helpful. So we'll start just with the first 3, I'll deal with all those together. Really when I started to get ready for this I mean, Professor Phillips and I have been talking about this problem for, I don't know, 2 or 3 years now, but we like to talk, and although the immunology work's gone ahead, I don't think that I've really have still grappled with the idea of how you actually move this on. But I think what I have seen is looking at the web over the last few weeks really is looking at the number of practices. Some of them are privately funded by private companies, some of them are funded by people getting their genetic tests, and some are actually funded by MSACs, the reimbursed tests. And there's a lot of people getting a lot of money out there for these tests. And so I want to know, how has that actually improved patient care? So have these groups that are getting the money and doing the testing, have they actually gone back and looked at how much contribution their testing has made to clinical practice? And I actually found a big fat zero, but I might have missed it. But I did a pretty thorough search. Apart from the areas, some of which we've already talked about, a lot of this other genetic testing doesn't actually come with any evidence of improved patient care. Now that's not to say it doesn't, but I think it is, if we are going to put a test up and we are going to get government funding, or patients are paying for it thinking that they're helping themselves, it is actually, it's on our shoulders that we need to be able to say to them, by having this test, we can actually reduce your adverse event risk by half, or we can actually, we'd like you to explain some of the side effects that you're having, but this information needs to be taken back to your general practitioner. And then just some examples of where it may have led to worse care. So really just to ponder this question, I don't have any answers. But as a clinician, I'm quite intrigued. A colleague of mine runs one of these, and I said to him, so when a patient comes to you and they've got this side effect that they're feeling a bit confused and they think it's related to their test, and they come to you and say, please run my P450 genetic test to tell me if I should be off this drug. I said, do you take a history? Do you sit down with the patient and talk to them about all this? Do you ask them what other drugs they're on, whether they take drugs and alcohol? Do you do a clinical examination routine, bloods? Or do you measure concentrations? Because we know we've got concentrations that actually correlate really well with toxicity and efficacy. No, none of that. The patient comes in, my clinician takes the blood, and then I give a computer readout to the patient. I say, oh yeah, okay, what does the patient do with that? Oh, I'm not sure. They don't know. Well, I can tell you what happens. And probably Professor Phillips can too. These people look up the AMA list of clinical pharmacologists around Australia and ring them. Because I've had this gene test, and I've got a poor metabolizing form of 2D6, and I'm on sertraline. Can you please tell me what to do? And I think, yeah, okay, how old are you? And I'm having this sort of phone conversation, which usually takes about 45 minutes. Then I have to ring the GP. Then I have to go and do some work to find out whether the drugs they're on are likely to be affected by all this. And I have to ring the patient back and say, well, actually, it's likely that the adverse event you've got is probably because you've got high concentrations of drug in your body, but it sounds like your depression is actually better. So you probably have to go back to your GP and discuss with them whether staying on this drug at this dose is actually a trade-off for the efficacy you're getting. And the patient's on the phone distraught. They've spent $750 or $1,250 doing this test. They then have to go back to their GP and then the psychiatrist. Now the GP could probably already say, well, you're doing all right from your depression, but actually I'm a bit worried about how sleepy you are, and therefore we should probably half the dose of the drug. So I'm just sort of worrying about this because we can't really measure what's happening, and I don't know why we can't have this information. It's a publicly funded test, so as taxpayers we're funding it. So why don't we see what the return on our investment is? And I'm actually going to remove this slide that's all right before we make it available, and I took the name of this company out here and here. But this is what it writes on the web. Through the use of the innovative genetic test DNA dose, so I look up the tests, I'm thinking, really, is that innovative? We were doing that in the 1990s in Christchurch, you know. It's a DNA test. Not really that innovative. We're able to analyse the profile of a patient's genetic variation. Fantastic. Well, actually, what they do is they measure 10 or 15 of these patients' pharmacogenetic variables, the ones we know about. And I think we do 5 2D6 mutations, not the 100 that we currently know are available. And then it maps this to the optimum drug and dosage for specific treatment. Now this particular patient that rang me, the one I was talking about with sertraline, there was no mapping going on. They just had on their readout people that are poor 2D6 metabolizers should think about, should reduce their drug. I can't remember the exact words, but it didn't actually map anything, which is the generic statement. So apart from 2 examples, this information actually does not exist. Now I've got here, doctors using this service have welcomed the clinical guidance. And 3 of us have got together about this whole issue because we were getting a bit upset with the time it's taking us to provide free information to patients and medical community when the person making the money was not actually not having to deal with any of this. So we don't feel that we haven't welcomed any of this. Patients don't feel relieved and empowered. This lady's actually crying. She was quite distressed about the amount of money it cost her. Some of it was reimbursed actually by the government, not all of it. The doctors and medicine specialists feel a lot of unpaid work is being created for them and the system. Patients are anxious as nobody can provide the guidance they require. That's I made that up, that weird stuff. So I think we have to, I mean, okay, I'm generalising, and the people I've spoken to are probably the people that are getting the brunt of it, but I think there are problems with it. So I also came up with about 5 references into the Sydney Morning Herald saying this pharmacogenetic testing could save millions. And I actually got back and been saying it's since 2008, and I'm starting to wonder when the millions are coming. We had a patient the other day who had this testing. He was on clopidogrel, and he had heard that some patients, so clopidogrel is a prodrug. It has to be converted to its active metabolite to be effective in stent thrombosis. It's converted to effective metabolite by one of the SIP here. And retrospective evidence has suggested that if you have this mutation, that you have an increased risk of new stent thrombosis. So we're not sure whether that tracks with the disease or whether that is actually a drug problem, but there's certainly a theoretical reason why people that have got this mutation actually get stents with clopidogrel. But this patient had been advised by his GP to switch to aspirin. I'm not sure why he wasn't on aspirin anyway to start with, because he had a 2C19 mutation, and therefore the cardiologist felt that this patient would benefit from clopidogrel. Now, I actually think that's really bad medicine. I mean, probably they need to double the dose of clopidogrel. Would have been the answer I would have given, but I actually had no evidence to guide that either. But before that, we would have just given clopidogrel and aspirin, and just the risk of in-stent thrombosis is so small now anyway that I wonder anyway whether we've just caused more of a health problem than what we're trying to prevent. This is another one that I was peripherally involved in through the group that Professor Phillips has already talked about in the UK, Professor Anne Daly. Her group showed, sorry, her group didn't show, her group noted that someone else had published that patients that had 2D6, the mutants of 2D6, sorry, that were fast metabolizers of the CYP2D6, tended to have a better outcome in treatment of breast cancer. And it's probably, well, theoretically anyway, because tamoxifen is also converted to its 100-fold increased potency metabolite endoxifen. Sorry, it binds with a 100-fold increased affinity to the estrogen receptor. There's quite a lot of literature actually on this, and the story is certainly believable. But more recently, there's been quite a lot of work showing that actually it's not a cut and dry story. And it's probably because there's over 100 mutations of CYP2D6, and there are other things that contribute to the metabolism of tamoxifen to endoxifen. And more important is the concentration of endoxifen. So if you can get high concentration of endoxifen, you certainly have a much better overall survival. But the 2D6 genotype only really explains some of the outcome. And this is also likely because if you're actually quite an obese lady, then your concentrations from the same dose of tamoxifen are likely to be quite a bit smaller, quite a bit lower. And if it is really related to the endoxifen concentration, then even if you're a poor 2D6 genotype, you actually won't get as high concentrations of endoxifen as a leaner woman. So actually it's the endoxifen concentrations that are much more important than whether or not you've got a gene mutation. And we do know, for example, that there are higher concentrations, that cancer outcomes are worse in obese people by 30%, even after you adjust for all these other factors. So it's quite likely that for a obese woman you need to measure endoxifen concentrations, not 2D6. This is another one that I'm a little bit disappointed over because I invested heavily in this. Warfarin. Warfarin gave jobs to loads of clinical pharmacologists in Australia, and it's a key part of our pharmacology curriculum, so we actually don't really want to lose Warfarin. So I worked for a long time looking at warfarin and hoping that these mutations of the metabolic enzymes that break down warfarin could actually help us explain why there's so much variability in warfarin. And we did actually find that there were some genetic mutations that seem to be associated with warfarin concentrations in the surrogate of the international normalised ratio. But when someone actually did the proper study, where 1,000 patients were randomly assigned to receive warfarin doses, either by using clinical data and genotype data or genotype data alone. Clinical and genotype or clinical alone. There was actually no difference to the time taken to get into therapeutic range, the combined outcome of an INR of 4, thromboemboli and major bleeding. Now, if you go back to the literature, there's about 10 years of genetic publications, and we're also very, very hopeful. A meta-analysis, and actually Professor Davey from the UK presented at the same time in the New England Journal, and Sturiopoulos has actually now done a meta-analysis to put it to bed forever, that really the genotype-guided dosing for warfarin is actually no better than the clinical algorithms. I next want to talk on the very few examples where we know genetic knowledge to guide dosing has actually improved outcomes, the ones we have evidence for. And maybe I'm a bit harsh on this, but I chose the ones that I thought had the best evidence. And actually we've probably already discussed these, so maybe we can just move on from those. But there's certainly cost effectiveness data showing how useful, how cost effective it is for the nation if we've got a properly validated test that helps us use the drug in a patient group that will get efficacy, and it stops us using it in a group that gets toxicity, and also reduces this issue, which is the compliance. So there is actually evidence, which I'll show in a minute, that improved patient compliance has occurred because of the testing. So people aren't afraid to take their pills because they know that if they're not the B57 genotype, then they are very unlikely to get this problem. Actually, I'll probably pinch this from you, Professor Noel. I think it's really good. Actually, this is quoted in the Deloitte summary of pharmacogenetics. So you can just look at the improvement in compliance. So before genetic screening, 15% of patients weren't taking their abacavir properly, and we know with HIV that you do need to have to suppress the viral replication, so you do need people to be taking their drugs as prescribed. And after genetic screening, I mean, this is all 2005, so it's probably zero now, but I think that really demonstrates the point of having a very well validated genetic test. Okay, and I think just the only point about the carbamazepine was just to really be clear of the effect of ethnicity, both on the presence of the gene, Weinberg law, etc., and also on the expression of the genotype. And this expression of the genotype is where I think pharmacology can make a real contribution, because we know there's so many factors that are involved there. So I think these, the successful translation of genotypes into clinical practice have been where a certain number of things have happened. You need, I'll put warfarin there. That did have a strong rationale, that's what I mean. I think we were, and we did have a very good surrogate that correlated with outcome. I think we've just been very disappointed by this one, actually. But key genes were identified that were very clearly correlated with outcome. Now, and we know that the outcome is related to the drug. And some of the issue that we have with the cancer work, I think, is that we know that the gene mutation is associated with poor outcomes, but we don't really know the effect of the drug and the gene combination on outcomes. We wanted to know about that, and some of that evidence is available in the clinical trials in the phase 3 study, but unfortunately for a lot of those big studies, after 8 or 12 weeks, as soon as the curves started to diverge, the non-treatment arm were allowed to cross. So we never really know for all those drugs how much better they are than current care. I can talk about that point later if anyone wants to. Validated testing and choice of population groups prospectively, blinded studies, and again that's validated post-hoc. So we go back into the data and then validate how many patients we need to treat, what sort of population statistics, what sort of phenotype data made this test very helpful. This is really sort of the crux, I think, of where a pharmacologist can make a difference, why knowledge of phenotype is important to medicine response. And I think if I can just, maybe I feel like, maybe I could send this to the NHMRC. Phenotype includes how one responds. So the genotype is a marker for what your genes say, but this is such a complicated area. And I think because it's so complicated, we will be needing to have mathematical models to be able to predict and weight some of these factors. A phenotype is based on basic pharmacodynamic and pharmacokinetic principles of drugs. That is to say, how the drug binds to its receptor, its affinity to the receptor, whether it can find the receptor. I mean, if you've got a really big, long monoclonal antibody, and we know that it blocks this receptor, but it can't actually get into the breast cancer because it can't get across the peak glycoprotein, for example, in the membrane pumps the drug out, or it's too large to actually diffuse across the membrane. That's kind of a problem, even if you know your drug does actually block the gene that we're looking at. And based on pharmacokinetic parameters, so how the drug actually moves around the body, again, if you've got a lot of fat around your gut wall and you're taking an orally absorbed drug, then the amount you're going to have actually on the other side of the gut wall is much less than someone who's lean. And for some of these drugs, if you take grapefruit juice or if you take other medications, you're going to really affect the absorption of these drugs. And just on that point, we know with some of the nibs, the imatinib family, they're not imatinib, you can actually increase the exposure of the drug from oral absorption by about 2 to 5-fold. Some have actually predicted more. So if you have a fatty breakfast, for example, fish and chips for breakfast, as opposed to cereal or milk, you can actually increase, you can double your absorption of the drug. This is phenomenal, really. And when you look at the drug information, so I go and I have a look, I wonder what the drug company said about that. And it says, take your drug at the same time every day. It doesn't say take it with the same breakfast. The same breakfast that was used in the drug company study. Okay. Yeah. All these factors, they're all common things that we know about, and some of them are governed by genetic processes and genetic markers, but a lot of it is to do with ethnicity, our body size, all the other drugs you're on, other diseases we have. And if I just pick on, I'll pick on old people in a moment because I think that's where I see most of the problems. Just to remind everyone, this fantastic thing. Adrian the other day came up to me and I was asking about pharmacology and he says, it's admin or, and I said, oh, okay, what's admin? Well, admin, it's in MINS. You didn't know what admin was. Admin is absorption, distribution, metabolism, excretion, and it's often the only information that you can get, and they're easy to find guides. So it's important to understand that some of these are governed by genetic processes, but some of these are also related to just who you are and your specific phenotypic variables. And that can decide whether or not a drug actually gets to a tumour, whether a drug actually gets to an infection. Pharmacology includes the response of the drug at receptors, concentration-effect relationships. What a novel thought, concentration-effect relationships. Here when you see these new monoclonal antibodies, the clinical trial data, do you see concentration-effect data there? Do you see dose concentration data? No, because a lot of it isn't actually there. And some of these have got through a fast-track FDA process where we've used a specific dose in a population group. So we don't actually know that, well, we know as pharmacologists that there's a huge variability in a lot of drugs between dose and concentration. But this information seems to have been importantly forgotten. And I don't know where you find that in the drug company clinical trial data package. I can't see it, but I think it's because most of these patients aren't on other drugs in the clinical trials. And they have specific other variables that mean that it's very difficult when you use this data in the real world. So this is my old man. My old man has reduction in lean body mass and total body water. So that means that if he's on a hydrophilic drug, he's got completely different pharmacokinetics and there'll be much different concentrations for him than a younger person. He's also got this increase in body fat, and all that body fat is actually around the visceral organs, which will affect the absorption across the gut wall. It's an oral drug. increased half-life and time to reach a steady state serum concentration. Now I think there is some new data in the cancer area in particular, and also in antimicrobials. But getting to the steady state it is often really, really important. You don't want resistance to develop on the way up if the drug's taking, a week to get there. We know obesity and undernourishment affect toxicity and drug failure, and all these facts which are common in our population in the hospital, they all affect drug absorption. All these diseases are also very common, and most of our patients have renal impairment. So you can see that really we've got a problem when we're trying to use these drugs with just genetic information. Okay. This is really to remind us that there are drug interactions, that there is drug disease and drug-food interactions. And I just highlighted here the summary information really that these are really commonly used drugs, particularly for patients in hospital, and they're often used in patients that are on cancer therapies. Now people think that, for example, an antacid or a PPI, oh well, that doesn't really cause any problems. Well, actually, a lot of the drugs that we use, particularly the NIBs, are dependent on the pH for drug absorption, so it is actually kind of a problem. And I think we shouldn't forget these issues as well, that we need to acknowledge comorbidity on drug outcomes, and this is what I see in general medicine. These patients come in under general medicine and they've been started on a therapy somewhere else, and these are the issues for them. And they're very significant. A lot of these people don't go home. This is very old data, but right back from about the 1990s, we know that comorbidity by itself has had a detrimental effect on cancer outcome. I'm glossing over the ethnicity, but because we've covered that. There is some work from Patricia Valerie from Menzies on the Indigenous population. How many genetic databases do we have in Australia on Indigenous people? How many Indigenous patients are included in the cancer clinical trials? I could bet 0. Okay, how many Indigenous patients have cancer? Well, actually, there's more cancer and there's a worse survival in Indigenous patients. So even after you adjust for all these other factors like time to late stage diagnosis, etc. So I think this woman is absolutely onto it and she's doing great things. But that's not incorporated if we're just looking at the new omics framework, the genetic testing, blah, blah, blah. We need that, but all these other factors and these people need to be involved in looking at things. And as my last point, I really wanted to talk about the phenotypes that we know that affect the relationship of drug to outcome. And again, I'm picking on rituximab because this was a specific issue of concern for us in Queensland. We started using rituximab in older patients, older women who had these haematological malignancies that ought to have benefited. We don't know what drug to use, but we know that we could completely wipe out all the B cells for about 3 years with the standard recommended dose. And they got a lot of toxicity and infection, so it was quite a problem. I know from sitting on high-cost drug committees that there are patients that for which we have given drugs for which they are not funded, that is to say they have KRAS negative colorectal tumours that have responded to EGFR receptor inhibitors. So some of our clinical and funding decisions about, oh well, if you've got this mutation and you can have this drug, we're probably denying patients who would benefit but haven't got the mutation, and we may well be giving, well, we know we're giving it to patients who are not responding. So there's something too simplistic about saying you're KRAS positive, you will respond to this drug. Should be you're KRAS positive, go and see the clinical pharmacologist, and I don't know what, no, I don't really need any more work, but we need someone to think about how to advise, how to contribute some drug information about this patient. Okay. This question is the one I really want to answer. What do we adjust for and how do we dose adjust these drugs? How do we get that information? Some of you have the information that's certainly included in some guidelines, but we don't have it all. And this is just to remind us that obese women with breast cancer had a 30% worse survival after adjustment for all these other factors. Now, if you flipped around and said what cancer drug has actually given us a 30% increase in survival in the last 15 years? None. So, in a way, with the genetics, we kind of lost the focus that actually we need to have our breast cancer woman normal body weight because they'll actually have a 30% better overall survival, hypothetically anyway. So I'm bringing up as a point to say we've been focused on this genetics of improving cancer outcomes, but we've forgotten about some of these other basic phenotype changes that are really affecting our cancer or overall survival. Okay, I think in conclusion I would just like to say that I think we really need to have a good understanding of particular patients that are receiving not just cancer chemotherapies but other new therapies in the population. We need to be linked in with a group that can help us understand how the clinical trial data really correlates with the patients that we've got in the hospital, to help us estimate what the dose should be for a particular patient with renal failure who wasn't in the clinical trial, to think about how the genetic information should be used in a framework to plan this patient's, for example, cancer treatment. These special groups of obese people, Indigenous people, different ethnicities that weren't represented in clinical trials, we need to think about that. I think we do need to have a knowledge of pharmacology, and we need to have reference to people that can help us in that area. And I think it is time really just to share the focus from PM, which could be personalised medicine or precision medicine. I think the flavour of the month is precision medicine, and I'm told that that's the geneticist's term, and as a clinician I'm allowed to use that. So I'll say personalised medicine to collecting real-world cancer outcomes to inform both the choice of drug and the dose. And I think we need to develop awareness of these factors and develop some collaborations.